Hello, everyone, and welcome to today's keynote presentation titled The Integration of Interoceptive Signals and Defensive Behaviors via Neurohypophysial Hormones. This webinar is part of the 7th annual event in the Drug Discovery and Development Virtual Event Series. I'm Christina Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that today's event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find that live chat located on the left of your screen. Now, if you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. And finally, as a reminder, today's presentation is educational and offers free continuing education credits. Click on the continuing education credits image located at the right of your screen and follow the process to obtain your credits. I would now like to present today's speaker, Joanna Dabrowska, Associate Professor, Center for the Neurobiology of Stress Resilience and Psychiatric Disorders, Department of Cellular and Molecular Pharmacology, the Chicago Medical School, Rosalind Franklin University of Medicine and Science. For a complete biography on our speaker, please find it on the left of your screen. Joanna, welcome. You may now begin your presentation.
Hi, everyone. Thank you very much for this kind introduction. My name is Joanna Dabrowska, and it's my pleasure today to talk to you about the integration of interoceptive signals and defensive behaviors via neurohypophysial hormones. So first, I want to start what are those neurohypophysial hormones? So we know that the neurohumoral system of the hypothalamus is the system of the nuclei that coordinate fluid balance and reproductive function. And this is happening via the action of the two peptide hormones, vasopressin and oxytocin. So vasopressin is this primary regulator of the renal water balance and blood pressure, whereas oxytocin is primarily involved in parturition and lactation, as well as maternal behavior. But also you will see from my talk that there are a bunch of other behaviors, so those vasopressin and oxytocin peptides are involved. So those two neurohypophysial hormones, oxytocin and vasopressin, are very structurally similar. So you can see that they are both nanopeptides. They only differ by 2 amino acids. And even though they do act on their own receptors, so oxytocin receptors for oxytocin peptide and vasopressin receptors for vasopressin peptide, because of these structural similarities, they are pretty promiscuous in their actions, such as they can act on each other receptors. And from those receptors, the oxytocin receptors and the vasopressin 1A receptor are the primary receptors expressed in the central nervous system in the brain. So there will be some crosstalk between those two peptides in the actions in the brain. So for example, oxytocin. So oxytocin is the peptide hormone and a neuromodulator that is produced in the distinct nuclei in the hypothalamus, including the paraventricular nucleus of the hypothalamus, the PVN. And we know that oxytocin from the PVN so is released to the blood circulation, and then on the periphery, it mediates crucial physiological processes like uterine contractions during labor or milk ejection reflex. But we also know that distinct nuclei from those hypothalamic neurons, they send projections to the central nervous system, including the limbic system. And we know that oxytocin in the brain mediates these really complex behaviors, including a lot of social behaviors. Those are the maternal behavior onset, the alloparental behaviors, partner recognition, social recognition, and so much more. And you can also appreciate that oxytocinergic fibers are reaching many brain structures, such as the central nucleus of the amygdala or the extended amygdala in general, that mediate variety of different defensive behaviors, including fear and anxiety-like behaviors. So in the way, oxytocin, because of its promising effects, using animal models and human studies showing the anxiolytic-like and anti-stress effects, oxytocin has brought a lot of interest as a potential pharmacotherapeutic for anxiety disorders or stress-induced psychiatric disorders, like, for example, post-traumatic stress disorder. So we know that it has been heavily tested for a variety of those disorders. We still, however, do not understand very well all of the cellular substrates and the cellular mechanism of these oxytocin actions where mediating fear and anxiety-like behaviors. So how do we conceptualize these defensive behaviors, and why do we even need fear and anxiety-like behaviors? So we know that we all possess this whole variety of defensive behaviors that really are designed to mobilize us in the face of danger. So we can detect a potential threat or an imminent threat, and we can, of course, react appropriately as well. So, for example, if you are hiking in the forest, you definitely need your adaptive fear, because you might need to have situations and you need to detect the threat, you might need to react to it. So you might freeze in some situations, you might startle or run away in some other situations. Whereas when you think about anxiety-like behavior, so in terms of the symptomatology of anxiety and fear, some of those symptoms can be pretty much indistinguishable, so they can be very similar. However, when we experience anxiety or anxiety-like behavior, we are usually talking about a potential threat or about something that can be more distant or more sustained. So basically what it means is that we experience anxiety, which can manifest as fear-like symptoms, but they happen beyond the immediate threat. They can happen in apprehension or anticipation of the threat, or more of a rumination of a threat. We also know that both of those anxiety and fear-like responses are adaptive, because we need to be able not to react to immediate threats, but we also need to be able to detect and react to potential threats. So that's why when we think about those defensive behaviors, we think about fear and anxiety as being different phenomena. So in a way that we think about fear, we wanted to conceptualize it as a selective response to a present or imminent threat. And this is associated with fear discrimination. So the way we understand it is this is a response to a selective threatening stimulus. Whereas when we think about anxiety, this is more of a response to a threat or potential threat, or in anticipation or rumination of a more diffuse, uncertain type of threat. And of course, this is more associated with fear generalization. And what it means is that there will be a response to a specific stimulus, but there will be also responses to stimuli that look kind of similar as the original threatening stimulus. But we also know that, of course, there is a continuum between those two types of responses. And why is this important? This is important because even though both fear and anxiety-like behaviors are adaptive responses and that are absolutely necessary for survival, we also know that some of those maladaptive fear processing can happen in anxiety disorders or stress-induced psychiatric disorders such as post-traumatic stress disorder. So, for example, in PTSD patients, the symptomatology that we observe is related to hypervigilance. So there can be a consistent vigilant monitoring of the environment. There can be a consistent increase in the so-called startle reflex. There is also an inability to discriminate between the cues that can specifically signal threat versus cues that signal safety. There is also a disproportionately higher fear reactivity to unpredictable versus predictable threats, and oftentimes deficits in the ability to extinguish this originally learned fear. So this, for example, this particular study was done by Christian Grillon group from NIMH, in which they look at the PTSD patients and have shown that when you do this kind of fear conditioning, basically paradigm in humans, and you use either predictable versus unpredictable threats, subjects suffering from PTSD have these significantly higher fear responses when the threats are administered in an unpredictable manner. So we know that central to the modulation of fear and anxiety is the extended amygdala. So this is primarily consisted of the central nucleus of the amygdala, as well as the bed nucleus of the stria terminalis, BNST. And according to this original classic theory of fear and anxiety formulated by Mike Davis and David Walker, the central amygdala is necessary for the short duration fear responses. So like a short duration cue fear responses, sometimes referred to as a phasic fear. So there's a very kind of discrete, specific type of threat. Whereas the BNST, it's more required for the fear responses to unpredictable, unsignaled, diffuse, or more ambiguous type of threats. So therefore, the central amygdala is primarily associated with higher fear discrimination, whereas the BNST is primarily associated with the higher fear generalization. However, we also know that those two structures are heavily interconnected via GABAergic and peptidergic projections. We also know that both of them project to a common brainstem autonomic center, so kind of effector centers for the fear responses, modulating cardiovascular respiratory function, but also freezing responses, startle responses. So in a way that even though those are primary output effects of those structures, we know that they do modulate each other activity, and in fact they both can modulate both fear and anxiety-like responses. So in the human functional imaging studies, for example, the BNST has been shown specifically activated in the condition of uncertainty during the anticipatory anxiety, for example, if patients suffering from a variety of phobias, during the hypervigilant friend monitoring. But what is really important and relevant for human psychiatry is that the activity of the BNST has been also shown to be increased in anxiety disorders as well as in post-traumatic stress disorder. So really understanding its function and the neurobiology of this structure and also the fear responses mediating by this structure is really can get us closer to understanding the neurobiology of PTSD better. So we know that in both humans and rodent models, the BNST is a predominantly GABAergic structure, so kind of containing a variety of inhibitory neurons that also co-express a vast diversity of peptidergic neurons, including some of the stress hormone neurons and a variety of other types. We also know that BNST receives strong projections from the hypothalamic oxytocin neurons. So we have shown before that PVN oxytocin neurons project directly to the BNST. And we also know from a variety of studies that BNST has a really high expression of oxytocin receptors and has a high density of oxytocinergic fibers. But we didn't really know much about what is the role of oxytocin receptors and what is the role of the oxytocin release in the BNST in mediating those fear and anxiety-like behaviors. So how do we study fear in a laboratory? So what we can use what we have been using traditionally is so-called Pavlovian fear conditioning, in which an animal is trained to associate a specific cue, so it can be a visual stimulus or auditory stimulus, with an aversive stimulus such as food shock. So then once this associative memory is formed, when an animal is then presented to this cue that was previously associated with that food shock, then it presents with the fear-like responses to the cue alone. And this can be evoked by the presence of the cue, so-called conditioned stimulus, but it can also be evoked by the environment in which the conditioning took place. So this is more referred to a contextual fear conditioning. So the way we do measure fear responses, and we kind of use this fear conditioning paradigm in the lab, is with the fear potentiated startle, so the FPS for short. So when you think about the FPS, in the FPS, an acoustic startle reflex is significantly potentiated by the cue. So again, it can be a visual stimulus, like in this case, that has been previously paired with the food shock. So you probably think about what is this startle response. So the startle response is basically what we all experience. It's a whole body jump as well as an eye blink that is evoked by a sudden unexpected noise, or maybe someone approaching you and unexpectedly tapping you on the shoulder. So we know that this startle reflex, this kind of a jump response, is significantly potentiated when an animal is fearful or as a human is fearful, and especially in the presence of the cue that has been specifically used to evoke the fear response before. So once we have those animals fear conditioned by co-pairing the aversive stimuli such as food shock with this specific cue, then after formation of the memory on the next day, we test those animals again in the presence of the cue. The way that we usually do that is we first test so-called the baseline startle responses, but just evoking those noises that provoke the startle response, and then we start presenting the cue. And we measure those startle responses or the fear responses during the presentation of the cue, as well as between the cue presentations. So what it really gives us is that the potentiation observed, so the highest jump, as you can see here, observed during the cue presentation is a measure of so-called cued fear response, right? So this is happening exactly during the cue presentation. Whereas what happens in between the cue presentation, we refer to as a non-cue fear. So that's the potentiation of the startle response observed between the cue that does not happen until after the cue is being presented. So this response, referred to as a non-cued fear, can be a measure of the cue-induced vigilance state, or more of a kind of an anxious arousal that is also associated with this fear conditioning. I spend a little bit more time on this specific slide because I wanted you to make sure that you understand the different components of the FPS, since we'll be also talking about them in a different experiments later on. So what we did first to understand the role of the oxytocin receptors in the BNST, we injected oxytocin receptor antagonists directly into the BNST before the fear conditioning in male rats. So then on the next day, we tested them for the fear recall using fear potentiated startle. And this was a drug-free test. And what you can see in the red circle is that in the animals that receive oxytocin receptor antagonists before the fear conditioning, we have a significant reduction of the cued fear responses. So that basically suggests that normally this oxytocin receptor transmission in the BNST facilitates the acquisition and the formation of the cued fear. Something that was also really interesting is that when we have measured so-called discrimination index, which is basically a ratio between the percentage change of the cued fear versus the non-cued fear, which is this anxious arousal observed between the cue presentation, again, with blocking those oxytocin receptors, we have a significant reduction of this discrimination index, again, suggesting that normally oxytocin receptor transmission can basically facilitate this fear discrimination in the via the BNST actions. So we definitely wanted to understand more about the cellular mechanism of these effects of the oxytocin in the BNST. So we turn into in vitro slice electrophysiology. So we know that there are at least 3 distinct types of the BNST neurons when it comes to the firing pattern and intrinsic membrane properties that are characterized into type 1, 2, and 3. We really know very little about the intrinsic and extrinsic connectivity, but we do know that the type 1 neurons are typically referred to as regular spiking interneurons in the BNST, whereas both type 2 and type 3 neurons can project outside of the BNST. And something that was really interesting to us is the fact that type 2 neurons, the great majority of them, project to the central amygdala. So it really got us intrigued because then we thought, okay, there's something really interesting happening between the BNST and the central amygdala interplay. in mediating those effects by oxytocin. So when we looked at the specific effects of oxytocin on the intrinsic excitability of those type 1, 2, and 3 neurons, what we have found was that oxytocin increased firing of type 1 neurons in an oxytocin receptor dependent manner, such as that the current evoked firing was increased after oxytocin application. We did not see this effect in type 2 neurons. We did see a trend in type 3 neurons as well. Now, we also looked at the spontaneous firing. So using the cell attached recording, we recorded spontaneous firing of type 1, 2, and 3 neurons. And something interesting that we found was that the great majority of type 1 neurons actually did fire spontaneously. And then when we applied oxytocin in those neurons, we've seen this increase in firing. Whereas in another type 2 neurons, we found that even though most of them were actually silent, in the neurons that were firing spontaneously, oxytocin was reducing their spontaneous firing. So this effect of increasing the excitability in one type of neurons and reducing the firing in another type of neurons made us think that there is something about the oxytocin effects that might also modulate the intrinsic synaptic activity in the BNST. So just basically modulating the intrinsic circuitry of the BNST in order to modulate the whole excitability of those neurons. All right, so we also use these recordings, the in vitro patch clamp recordings, to determine the role of oxytocin on the intrinsic excitability of type 1, 2, and 3 BNST neurons. And what we have found was that oxytocin increased the current evoked firing in type 1 neurons on the top left, an effect that was blocked by oxytocin receptor antagonist. We have not seen the same effect in type 2 neurons, so there was no effect. There was also a trending effect in type 3 BNST neurons, showing those excitatory trends on the intrinsic excitability. So to really look at the effects on firing, we also turned to the spontaneous firing. So this was done during the cell-attached recordings. And what we have found was that the great majority of type 1 neurons were firing spontaneously. When we applied oxytocin in those neurons, we actually seen an increase in the spontaneous firing. Whereas type 2 BNST neurons, most of them were silent. However, in the neurons that did fire spontaneously, when we applied oxytocin in those neurons, we did see a reduction in firing. So this was a really interesting phenomenon that we have seen increase in excitability in one population, but also reduction in the firing in another population, which made us think that oxytocin can also modulate the synaptic activity of the BNST. There can be something happening with the intrinsic circuitry modulation by the oxytocin peptide. So what we did here, we recorded synaptic events. So we recorded inhibitory synaptic events, specifically spontaneous inhibitory postsynaptic currents, IPSPs for short. And what we showed was that oxytocin increases the frequency of IPSC, spontaneous IPSC, specifically in type 2 BNST neurons. So the same neurons that we've shown the reduced firing, spontaneous firing. And again, this effect was observed only in type 2 neurons. It was blocked by oxytocin receptor antagonists, and it was also blocked by sodium channel blocker, TTX, tetrodotoxin, basically suggesting that the effect we are seeing in type 2 neurons, the effect on synaptic activity, is mediated or is driven by firing action potential of another population of BNST neurons. So it kind of made sense to us now that we have this effect on the interneurons, the excitatory effect, and then increased synaptic activity in type 2 neurons. We also recently replicated these findings basically using optogenetics. So in this particular case, we have expressed, so we use the AAV driving channel rhodopsin expression in the paraventricular nucleus of the hypothalamus under oxytocin promoter. So we are showing on the top right that we can evoke firing of these oxytocin neurons, which with each pulse of the blue light. But then we turn to the tetanic light stimulation in the BNST fibers because we wanted to be able to evoke oxytocin release. And we were able to show that in these type 2 neurons of the BNST, again, we see an increase in the frequency of spontaneous iPSCs. So this is something we've shown before with the exogenous oxytocin. And really interestingly, we could block it with the oxytocin receptor antagonist. So suggesting that, yes, we can evoke endogenous oxytocin release with optogenetics. So this was really interesting to us because now that means that we have this increased excitability after oxytocin in type 1 and potentially also type 3 BNST neurons, which then provide this inhibitory synaptic activity to type 2 neurons and then reduce their firing. Now, if you remember, those type 2 neurons have shown that the great majority of those neurons actually project to the central nucleus of the amygdala. So what we did next, we injected these retrograde tracers, so the green retrobeats, to the central nucleus of the amygdala. And we recorded specifically from the central amygdala projecting BNST neurons. So you can see these neurons, the green fluorescent neurons in the BNST, those are the neurons that project to the central nucleus of the amygdala. And when we record specifically from them, we are able to show again that oxytocin increases the spontaneous iPSC's frequency in those neurons. So again, suggesting that yes, oxytocin has this indirect inhibitory effect on type 2 neurons projecting to the central nucleus of the amygdala. So this kind of started to make sense to us because we realized that by inhibiting this projection, is basically GABAergic projection from the BNST to the central nucleus of the amygdala, oxytocin can disinhibit the central amygdala activity. And of course, as a result, it can behaviorally then lead to increased acute fear expression. But of course, we did want to test it behaviorally. So in this particular case, what we did, we used projection-specific chemogenetics. So in this particular case, we injected a retrograde tracer driving Cre expression in the central nucleus of the amygdala. And then we also injected Cre-dependent expression of excitatory dreads in the BNST. So then we had those dreads, excitatory dreads, expressed specifically in the BNST to the central amygdala pathway. So now with the dreads ligand like CNO, clozapine, and oxide, we can then manipulate those neurons specifically to test their effects and their role in the modulation of fear and anxiety-like behaviors. So this is what we did, and we tested those rats for FPS, for the fear potentiated startle again. And in this particular case, the CNO, so the ligand for dreads, was injected before fear conditioning. And when we first tested those animals on the following day, we have not seen any effect in acute fear expression or the discrimination index. However, when the animals were tested again on the following day, drug-free, for acute fear and a discrimination index, we see that the acute fear is significantly reduced. So basically, we see this reduction in acute fear expression, and we also see this reduction in discrimination index. So then the idea is that by exciting the BNST interneurons, type 1 and or type 3 neurons, oxytocin can inhibit type 2 neurons of the BNST, which project to the central amygdala. Now, by inhibiting the GABAergic inhibitory neurons, the projection neurons, it basically provides this disinhibitory effect on the central amygdala and therefore can increase acute fear expression. So for the interim summary of this first part of the talk, I wanted to tell you that oxytocin was shown to excite both type 1 and type 3 neurons of the BNST via oxytocin receptor. We also know that oxytocin receptor transmission facilitates acute fear and fear discrimination as measured in the fear-potentiated startle. Chemogenetic activation of the BNST to the central amygdala neurons reduces acute fear expression. And oxytocin receptor transmission in the BNST facilitates acute fear formation by disinhibiting the central amygdala. So we know that activity of those oxytocin receptor neurons in the BNST can reduce the expression of cue-induced vigilance state. And of course, these findings do have implications for understanding neurobiology of anxiety disorders and PTSD, and potentially providing novel targets for pharmacotherapy of anxiety disorders and PTSD. So now, switching gears a little bit, what about the sister peptide, vasopressin, which we know also projects to the extended amygdala from the hypothalamus? So there are distinct nuclei projecting to the extended amygdala, including the BNST, although we do not know the exact sources of those projections. We also don't know anything about the cellular effects of vasopressin in the BNST. So we did a similar experiment in which we switched to in vitro slice recordings of different types of the BNST neurons. And when we recorded from those different types, we showed that vasopressin, similarly to oxytocin, also excites type 1 neurons of the BNST. Now, we use variety of different antagonists before vasopressin to determine the receptor specificity. And we have shown that blocking oxytocin receptors before AVP can actually abolish the effect, whereas blocking vasopressin 1B receptor has no effect. So again, suggesting that similarly to what we've seen before with oxytocin, vasopressin has this direct excitatory effect on type 1 neurons mediated via oxytocin receptors. Now, we also saw a powerful excitatory effect of vasopressin in type 3 neurons. We use, again, a variety of antagonists. So oxytocin receptor antagonist was able to block the effect in type 3 neurons, whereas both vasopressin 1B receptor antagonists as well as vasopressin 1A receptor antagonists did not fully block the effect of vasopressin, suggesting that again, vasopressin excites those type 2 neurons of the BNST via oxytocin receptors. Now, knowing that, we switched to some of the potent agonists for those receptors, and we use a selective agonist for OTR, which is more powerful than oxytocin itself, TGOT, and we show this strong excitatory effect in type 3 neurons by TGOT. Whereas when we use the selective RAD vasopressin 1A agonist, we saw a pretty modest effect, again showing that the oxytocin receptor is a major player of these excitatory effects in type 3 neurons. Now, lastly, to confirm these effects, we also took advantage of some of the transgenic RAD models. So in this particular case, we use oxytocin receptor QRAT, transgenic RAT. So we recorded from fluorescent oxytocin receptor expressing neurons in the BNST. We were able to confirm that the great majority of them actually fall into type 3 category. And when we recorded the excitatory effects, we show that vasopressin has these really strong excitatory effects on those oxytocin receptor expressing neurons. Now, previous studies have also shown that the great majority of type 2 neurons in the BNST express CRF, so the corticotropin releasing factor, stress hormone mRNA on a single cell level. So we were also able to record from some of the CRF neurons, fluorescent CRF neurons from the CRF-CRE transgenic rat. And again, these neurons were all categorized into type 3 neurons of the BNST. And we have shown that vasopressin can have this excitatory effect on those type 3 CRF neurons as well. So now the idea is that if we have those oxytocin receptor expressing neurons excited by both vasopressin and oxytocin in the BNST, what is the role of these neurons in mediating fear and anxiety-like behaviors? So what we were able to do, we were able to take advantage of this oxytocin Cree rat, so in which the oxytocin receptor is expressed, basically under the oxytocin receptor promoter. So we have these fluorescent oxytocin receptor neurons, so there is a Cree expression in these neurons, and we can introduce some viral vectors, Cree-dependent viral vectors, to manipulate this neurons activity. So in this particular case, we use inhibitory dreads to silence, or Cree-dependent inhibitory dreads to silence those oxytocin receptor expressing neurons in the BNST. And again, we silenced them before fear conditioning, and then we tested them again for the fear-potentiated startle. Now, similarly to the previous experiment, when we tested them drug-free for the first time, there was no effect on acute fear. So the saline and the CNO group, the dreads ligand CNO group, have the similar effects of acute fear expression as well as non-acute fear expression. Whereas when we tested them on a following day to kind of test the extinction of those fear responses, whereas the acute fear was not affected, we did see a significant increase in the non-acute fear in groups treated with the CNO. So basically, this tells us that normally activity of those oxytocin receptor expressing neurons reduces this non-acute fear, so reduces the cue-induced vigilant state in those neurons or the anxious arousal, which is associated with fear conditioning. So now we also tested the same animals for the activity in the elevated plasmase. And what we were able to show was that when the rats are treated with the CNO, so when those oxytocin receptors neurons are silenced, then the activity in the open arms of the elevated plasmase goes down, whereas the activity in the closed arms is increased. So again, this suggests to us the activity of those oxytocin receptor expressing neurons is increasing normally the time spent in the open arms of the elevated plasmase in male rats. So again, suggesting, so based on those reduction in anxious arousal and increased time in the elevated plasmase in the open arms of the elevated plasmase, we can conclude that activity of those oxytocin receptor expressing neurons is basically promoting this anxiolytic-like behavior, so reduction in the anxiety-like behavior. So now, the fact that we're having these oxytocin effects and the vasopressin effects, and they are both acting via oxytocin receptors in the BNST, so they really get us very puzzled, because we were just trying to conceptualize it and just wondering why are those effects so redundant, right? We have those two peptides basically having very similar effects on those oxytocin receptor neurons activity. So then we realized that this is true, but also vasopressin and oxytocin are produced in some of the distinct nuclei of the hypothalamus. So we really switched to this tracing of some of the vasopressin neurons to better understand what is the source of the vasopressin in the BNST. And then maybe perhaps those sources of oxytocin and vasopressin coming from distinct nuclei of the hypothalamus could be in fact modulated by different physiological conditions, different internal states. And in fact, different physiological states can favor the release of oxytocin versus vasopressin in the BNST. So we started to map some of those projections using recently developed vasopressin Cree rat, in which, again, we were able to map vasopressin neurons in a specific hypothalamic nuclei. And what you are looking at here is the vasopressin neurons in the suprachiasmatic nucleus of the hypothalamus. So again, we are able to inject the Cree-dependent viruses, like in this particular case, the virus that drives the mRuby expression under also synaptophysin promoter, so it allows us to kind of track the axons and processes of those neurons. And when we really look at some of those neurons in the projections in the BNST, we could see fibers in the bed nucleus of the stria terminalis, so in the PVN, that also express vasopressin peptide. So basically suggesting to us that those suprachiasmatic AVP neurons project to the BNST, and they also co-express mature vasopressin peptide. So of course, we wanted to test it functionally. So is this projection also functional? Can we actually evoke peptide release using this projection? So what we did in here, we also used the advantage of the AVP Cre rat. So we expressed the channel rhodopsin under Cre in the suprachiasmatic nucleus of the hypothalamus. And we were able to use tetanic light stimulation to evoke vasopressin release in the BNST. Something that was really interesting to us is that we actually could see that there is an increase in the spike frequency if those type 3 neurons that we have shown before increase excitability after AVP. But when we pre-apply oxytocin receptor antagonists before the tetanic light stimulation, we do not see this effect. And again, showing on the right, this effect being blocked by oxytocin receptor antagonist. So this really got us very excited because, as you know, suprachiasmatic nucleus of the hypothalamus is this master circadian pacemaker, and especially vasopressin signal is the signal that really prepares the animal for the resting phase. So it really got us intrigued about how this preparation for the resting phase and the vasopressin release in the BNST can then interplay with some of those defensive behaviors changes when it comes to the circadian rhythmicity. We also look at the neurons, AVP neurons in the supraoptic hypothalamus, nucleus of the hypothalamus. And again, we were able to track some of those projections from the SON to the BNST, showing that they also do express mature vasopressin peptide. We also did functional experiment trying to record from those AVP neurons in the supraoptic nucleus of the hypothalamus after they express channel rhodopsin. So we are able to show that we can, in fact, increase their firing with the blue light pulses. And then again, if we use tetanic light stimulation in the BNST, we are able to evoke AVP peptide because we do see an increase in a fraction of increase in firing in a fraction of the BNST neurons, type 3 BNST neurons again. And this effect is again blocked when we pre-apply oxytocin receptor antagonist before tetanic light stimulation. And of course, because of the SON involvement in the regulation of water and electrolyte balance, again, this is a very interesting avenue for us to explore about how changing water and electrolyte balance and the activity of those AVP SON neurons can then again interact with the changing levels of defensive behaviors by directly impacting the excitability of the BNST neurons and as such the levels of fear and anxiety-like behaviors. So this is just a simplified diagram to kind of show that, for example, there is an interplay between the priorities for survival. So if the rodent is thirsty and there is a situation in which it needs to cross the meadow to get to the water spring, then of course there is this interplay between the need for water and then the levels of the defensive behaviors that we can engage in also to survive. So to summarize the entire talk, I did show you that both oxytocin and vasopressin excite BNST neurons via oxytocin receptors transmission. I also showed you that oxytocin transmission facilitates acute fear formation by disinhibiting the central amygdala, and that activity of those oxytocin receptor neurons reduces the cue-induced vigilant state, so the anxious arousal, and increases open arm exploration in the elevated plasmase, so overall leading to this reduced anxiety-like behavior. I also showed you that vasopressin fibers in the BNST originate from the suprachiasmatic nucleus of the hypothalamus as well as from the supraoptic nucleus of the hypothalamus. And just to be clear, we don't yet exclude other sources as well. I also showed you that evoked vasopressin release from the SCN and SON fibers excite those type 3 neurons via oxytocin receptors. And of course, overall, these results have implications for how changing physiological conditions such as lactation, circadian rhythm, water electrolyte balance would then modulate those defensive behaviors via those diverse oxytocin and vasopressin inputs into the oxytocin receptor neurons in the BNST. And just broadly speaking, it really also gives us avenues to investigate how those changing physiological conditions would then modulate defensive behaviors and anxiety, feel-like behaviors in anxiety disorders and PTSD. And with that, I would like to thank, of course, the funding sources, but most of all my colleagues and collaborators in my lab. So Fulvia Berton, Walter Francesconi, Rachel Chudoba for the great majority of the electrophysiological studies, Valentina Oliveira for her behavioral studies, which pretty much all of the studies that I have shown you today were run by her. Sue Olson for the beautiful hypothalamic injections, and of course all of the lab members for the hard and very inspiring work. Thank you.
I'd like to thank again Joanna Dabrowska for her time today and her important research. Now, before we go, I'd like to thank the audience for joining us today, and questions submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand for 1 year, and that's through February 21st, 2025. LabRoots will alert you via email when this webinar is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us and have a great day. Bye-bye.